trim it, then at that point you have one section of aligned DNA to do analysis. So it's a lot easier to get the looks of confusion. So after doing all this and after looking through what we had from the database and everything, we were able to construct a rarefaction curve, and this is based off of phyla, and at this plateau region at the top here, right up here, it's showing us that we have sort of reached a maximum number of phyla that we'll find in this area. If we didn't have that plateau, then we would expect that, we need to do more sequencing until we can get a total coverage there. And as far as the phyla present, we had proteobacteria, and 3 of the groups of proteobacteria subphylas, alpha, beta, and gamma. Planktonomycetes, geomethamonidates, actinobacteria. 2 significantly different unknown groups showed up, and Firmicutes, chloroflexia, and acetobacteria. And here's a picture of our bootstrap tree. I know it's going to be a little bit difficult to see exactly what's going on there, but I tried to sort of highlight where the different regions were separating out. And a chart showing how much of each of these we have. So as you can see, the proteobacteria pretty much dominated this particular sample, and followed by planktonomycetes and actinobacteria. But as you see these 2 unknown groups, they're having a pretty big number in there too, which I thought was kind of interesting. What I have here is I have a comparison of the samples that I took from the cave that I looked at, and some samples from some other caves and some other environments which were forest and agricultural, to try and make a comparison of these environments as a whole. Like, how different really are these cave environments from say a forest environment or an agricultural environment, just a soil sample that you would take anywhere. And as you can see here, there are some fairly distinct differences between these samples, and that would be that acetobacteria is showing up in these caves. However, it's not showing up at all, and here's the cave samples here, and it's not showing up at all in any of the forest or agricultural samples. And also, there's this, I don't know how to pronounce that. This phyla doesn't show up at all in the caves, yet it's in all of these forest and agricultural samples. There's also, a low number of the chloroflexes showing up in them. But really what I see from this is that there may be more difference between one particular area in a cave, so one particular environment or area within that environment, than there is difference really between, say, a cave versus a forest environment. That's, because if we look at this is this cave here, that sample was taken from pools that were fairly high acidity, things like that. So this sample is showing far less diversity than either of the other 2 cave samples. However, the other 2 cave samples are pretty similar to that of the forest and agricultural. And here's a rarefaction curve showing the phyla present. He's starting off with this being the one that I found the cave that I was working at. Here's a Spanish cave that was taken, 2 Spanish caves, and this is a compiled data from the forest and agricultural spots. And this was data taken from a cave in Lower King. And as you can see from this, the diversity of these is actually, it looks like there may be more diversity in some of these cave samples that are, equivalent to that of the forest and agricultural sites. So that sort of mentality that these environments are very, they're not very complex. There's not a lot of diversity in there. That has a lot to do with sampling techniques as well, because these people, the people that have done these previous studies, a lot of them, that have been using culture-dependent techniques. And culture-dependent techniques they lack in the ability to get a full coverage of the bacteria present. I've read that less than 5% and as low of a number as 0.1% of bacteria are actually cultural. So to do these strictly culture-based techniques, you're really missing out on a lot of the bacteria present, and that's being shown in these studies showing that there is actually a lot of diversity in these areas. So now I'm going to talk a little bit about the environmental roles that some of these particular phyla, and in the case where I was able to find genus or family or whatever matches through GenBank, then I'll talk about those specific genus or family as well. Proteobacteria was the most common, as I said. I found these families, and particularly one thing I'm seeing with these is that you're finding a lot of organisms that can fix nitrogen, and you're finding some pretty neat stuff going on with that. And specifically this nitrospiral, I think is pretty interesting. It's an ammonia oxidizing bacteria, and I think that it's a good kind of an indicator of some things. This particular genera of bacteria will show up when less than 1 millimolar of nitrogen is present in the sample, where if there's a lot higher concentration, certain other species will come into play and sort of, I guess, outcompete this guy. So that's sort of an interesting little note there, that's sort of an indicator of Firmicutes, specifically Bacillus genus was found in this. And Bacillus are known for being able to do several things in the environments. Really, if a bacteria can do it, there's probably somebody in the Bacillus genus that can do it. They seem to be a jack of all trades. But they're used in bioremediation, antibiotics, nitrification, nitrogen fixation. They have chitinase activity. So there's some of these organisms that can break down some of that chite that we were talking about earlier. And also beta-carotenases. And here's possibly one of those chains that we were talking about earlier. So they can anaerobically reduce this nitrite to ammonia. Well, we have another organism that can change ammonia into nitrite. So this may be one of those elements that are being shifted back and forth in the environment. This may be, this is very possibly one of these chains that are in there. Actinobacteria, these are as well used in biodegradation, antibiotics, anti-cancer treatments, anti-angiogenic cancer treatments, and immunosuppressive drugs. And these also have some species that show chitin-lytic activity. Some of these have antifungal properties as well. And what's interesting is these have been studied specifically to have antifungal properties to the genus Fusarium, which is causing that white nose syndrome. So, maybe there's a possible correlation between these bacteria and the infection, and that that's something that could be possibly looked into for some of these infected areas. Chloroflexi, it's traditionally thought of, it has a lot of photosynthetic species. It's been able to be used in the dechlorination of PCBs, and obviously at that point bioremediated. So this kind of brings up a couple questions, and some of these Planktonomycetes as well is thought of to be sort of associated with photosynthesis as well. So is there other ways that these organisms are gathering nutrients? Probably. They probably have several pathways that they can use to gather nutrients. If they're given the opportunities, they'll probably go through photosynthesis. If not, they probably have other pathways they can utilize. There's also the possibility that with the stream that's running through this cave, that these organisms are just being passed through with the stream. And the methods that we use, if the DNA is present, then we have a chance of picking it up. It could be dead organisms that are just flushing through the creek or whatever. They're still going to be present in the sample. So there's possibilities that things are flushing through and we're just picking them up as they're coming through. That doesn't make them any less, I guess, necessarily important in the environment, because there's still the chance that they still have activities going on while they're in this area.